Hi guys, lots of you requested it, so this is my all-in-one new spec biology AQA video, and it's long. I'm not sure how long it is yet, but it is very, very long. So I'm going to whistle through the whole spec, and hopefully you'll find it really, really helpful. As always, I add like perfect answers all the way through this video. If you want to get hold of a copy, because I've actually written that all up as a document, a PDF, just email me sciencewithhazel@gmail.com to get hold of one. Anyhow, let's get going. So let's start by looking at the microscopes. Remember, there are two microscopes you need to know about. They are the light and electron microscope. Get those sorted in your head as to what they are. The light microscope you find in your classroom. You've obviously used it in science lessons, I hope. Electron microscopes are the ones you find in big scientific laboratories, and they are very expensive. So in terms of comparing how well they actually work, look at the magnifying powers. While light microscopes only magnify up to 2,000 times, electron microscopes up to 2 million times. Then we'll look at the resolving power, which is the ability to distinguish between two separate points. Light microscopes can only distinguish between points which are 200 nanometers apart, electron microscopes 0.2 nanometers apart. There are good things about light microscopes. You can use living samples. For electron microscopy, they very much have to be completely dead. And light microscopes you can see in colour. Electron microscopes, it will be black and white, but you often see pictures which have been coloured in or dyed. Now I'm going to show you how to use the magnification equation. This is your formula triangle that you need, which is image size, magnification and object size. So obviously if you're looking for magnification, you cover that. So you see that it's image divided by object size. If you're after image size, then you just do magnification times object size, and for object size you just do image divided by magnification, and I always use this triangle here to help me. Remember, when you're subbing in your numbers to the formula, you've got to use the same units, otherwise you'll screw up. So I recommend converting everything to millimeters. So to do centimeters to millimeters, you take your number that's been given in centimeters and you times it by 10. If you've been given the number in micrometers, then you want to divide it by 1,000. I'll show you an example now. So the image of a cell in a book is 48 centimeters in length. However, the real cell is only 120 micrometers. Calculate the magnification. So I'm going to use the triangles to tell me that magnification is image divided by object. Then we need to get the image size into millimeters. So I've been told it's 4.8 centimeters. So according to my writing in black, I need to times it by 10 to get it into millimeters. So it becomes 48. And then we need to convert the 120 micrometers, which is fuzzy over here, into millimeters by dividing 120 by 1,000, which is how I've got 0.12. Plug that into your calculator and you get an answer which is a magnification of 400 times. Let's look at another question. A student measured the diameter of a capillary in a micrograph. The image measures 5 millimeters and the student knows the magnification is times 1,000. How many micrometers is the diameter of the capillary? Using our formula triangle, then we're after the object size, so we're going to do object size equals image divided by magnification. So I've written the equation here, and then we're just going to pop those numbers in as they are. So we're going to do 5 divided by 1,000, which gives us a value of 0.005 millimeters. However, it wants the answer in micrometers, so therefore we need to do the opposite of my conversion. So we're going from millimeters to micrometers this time. So obviously we need to do the opposite of this, which is times by 1,000. So that brings the answer to 5 micrometers. So first of all, our animal cell, remember it looks like a fried egg basically. It's got a big border and then it's got a small compartment in the middle. Remember that compartment is the nucleus. The nucleus controls the activities of the cell, so there's your key definition. Then you have some jelly which surrounds it, and we call that the cytoplasm. Role for that is that it's where all the chemical reactions take place, so that's another key definition. Surrounding the cell you have a cell membrane. Please note that there is no cell wall in an animal cell. The only thing you have is a border, a single border, which is the cell membrane, and that controls the entry and exit of substances inside the cell. Then you'll have to learn mitochondria. That's really the powerhouse of the cell, and it creates energy via aerobic respiration. Remember, aerobic respiration means that it requires oxygen. Then you have some tiny dots. These are ribosomes, and they're responsible for protein synthesis. Moving on to the plant cell, you find exactly the same structures in the plant cell. Nothing's missing, you've just got a few additions. And first of all, make sure you're happy that a plant cell is very rectangular in shape. and that's because surrounding it is the cell wall, and that supports the cell. The rectangle in the middle, called the vacuole, that contains cell sap. And then we've got a nucleus squeezed into one corner, again like the animal cell, that controls the activities of the cell. We also have mitochondria, but crucially we have some green things which look very similar to mitochondria, but the fact that they're green tells us that they're chloroplasts, and they're full of a pigment called chlorophyll, which gives it its green colour. That's where photosynthesis takes place. Bacteria are responsible for some of the most horrendous diseases out there, things like tuberculosis. They're bacterial diseases. Cholera is another one. This time you've got more of a distinct cell. You have a cell wall. You have a little tail, and we call that a flagella, and that helps the bacteria move to where it wants to go. Sometimes you'll see diagrams with a capsule around the edge or a slimy coat. That's just an additional structure. Don't worry too much about that. The crucial thing about a bacterial cell is that there's no distinct nucleus. What you have instead is a nucleoid, and then you'll see some tiny other structures, and these are plasmids, and these are also genetic information, and they're really important when we're talking about genetic engineering. Now we're going to talk about nerve cells, and I'll try and add a picture of a nerve cell because they have a very unique shape. Now, first of all, they have lots of dendrites, and that allows them to make lots of connections with other cells. They have a very long axon, which is the central kind of actual nerve that forms the length of the neuron, and what that does is that it carries the nerve impulse from one place to another. Because they're very active cells, obviously they need lots of energy, so you'll find that they have lots of mitochondria to produce that energy in respiration. And lastly, the nerve endings have special synapses, which are gaps where a chemical diffuses across in order to convert that electrical impulse into a chemical one. Now, some of these adaptations, I think you can see they're not really distinctive adaptations. It's more about the fact that you're able to describe the cell in detail. So I do recommend that you do shove in as many specialist words as possible. Now we're going to talk about muscle cells. Muscle cells contain special proteins which can slide over each other when the muscle contracts. Because they're very hard-working cells, they're constantly contracting, they obviously need lots of mitochondria to provide the energy needed for muscular contraction. And lastly, muscles can store glycogen. Remember that glycogen is the storage compound of glucose, and it's there so that when lots more energy is needed, this substance can be broken down in order to carry out respiration. Sperm cells now, I like sperm cells because they're really easy to remember. First of all, remember that they have a very characteristic shape. They have a whiplash tail which helps them to swim. They have a central portion which is packed full of mitochondria to provide energy to enable them to swim. They have a head containing genetic information. And lastly, they have some enzymes in their head which help break down the outer casing on the egg when they need to penetrate it. Those enzymes are found in a special structure called the acrosome, another very good specialist word. Now we're moving on to specialist plant cells. So we're going to start with the xylem. Remember, xylem transport water from the roots up the stem to the leaves where it's needed in photosynthesis. Xylem have very special structure. First of all, they're dead, and that means that they're hollow because they've lost their cell contents ages and ages ago. That hollowness means that obviously there's lots of space for water to be transported. They have special walls made out of lignin, and that lignin is a very strong substance which will help keep the xylem open. Now, coupled with xylem, you find that there are phloem. Remember, phloem transports sugars around the plant, so from the leaves where the sugar's made up and down the plant for storage. Now, you find that the ends of the phloem cells break down and form sieve plates, and those sieve plates allow the food to move easily in the water in which it's carried. Because phloem cells need lots of energy, they're coupled with companion cells. Now, companion cells are full of mitochondria which provide the phloem with energy. Let's take a regular plant cell now. So we call this the palisade layer if we're looking at the structure of the leaf, or the palisade mesophyll. Obviously they contain lots of chloroplasts. Why? Because it's full of a pigment called chlorophyll which absorbs light in photosynthesis. So that's really, really key. You do find that these plant cells, or these palisade cells, are located near the top of the plant in order to capture as much sunlight energy as possible. And because, as with all plant cells, they have a large permanent vacuole, and that helps keep the cells rigid. Lastly, we're going to look at root hair cells. So if you're asked to draw a root hair cell, basically draw a plant cell but with a big elongated area which is the root hair. And obviously don't include chloroplasts because it's a root, it's underground, there's no sun going to reach there, so it would look really stupid if you added chloroplasts. And just draw all the other structures as usual, your cell membrane, your cytoplasm, your cell wall, etc. And just point out that that root hair gives a very large surface area to allow all that water to be absorbed. Remember, it has a large permanent vacuole which helps speed up the water movement by osmosis, And again, they have lots of mitochondria. Why? Because they need lots of energy to actively transport minerals from the soil into the root hair. Now, your definition for diffusion is that it's the net movement of particles from an area of high concentration to an area of low concentration. This happens very readily. Things change when you're talking about active transport. This is the total opposite, and at this time the net movement of particles is from an area of low concentration to an area of high concentration, and this requires a lot of energy. I'm going to be covering osmosis, and I'm going to be talking about the effect that placing various types of cell in different solutions has on them, and I'm going to give you an overview of the definition and talk about keywords like plasmolysis, crenated, and all these sorts of things which sound hard, but I promise that they're not. So osmosis, crucially you need to know that it's to do with transport in water. So our definition is it's the net movement of water particles from an area of high concentration to an area of low concentration through a partially permeable membrane. Make sure you have partially permeable membrane in there because there will be a mark available for that. I'm going to talk now about the effect of placing plant cells in different concentrations of solution. Now remember that a plant cell differs from an animal cell in that it has a cellulose cell wall, a vacuole and chloroplast. So what happens if you place the plant cell in a very salty solution is you'll find that there's more water, so higher water concentration, higher water potential within the plant cell, and that there's a lower water concentration, lower water potential surrounding it. Therefore water, based on our osmosis definition, will move from the inside of the plant cell to the outside. This will cause the whole cell to shrink, so you'll get this very distinct shrunken appearance, and we call this plasmolysis because what happens is the cell membrane pulls away from the cell wall and you end up with this characteristic wavy line. So obviously this isn't a great situation for our plant cells to be in. If we turn this around and we place our plant cell instead in pure water, so that's with a very high water potential, a very high water concentration, there's obviously going to be more water outside of the plant cell compared with inside. So water is going to enter the plant cell this time by osmosis, and you end up with the plant swelling up and becoming turgid, which is where the cell wall kind of swells out and it looks quite balloon-like. This is a great situation for our plant to be in because when the cells are turgid, it means that the stem can support itself, for example, and the plant doesn't wilt. The reason why this plant cell doesn't explode is because of the existence of the cell wall, which helps support the cell and prevent the pressure of the water inside breaking the cell. So it's very important for plants. However, if you have a solution instead now that the plant is in that's kind of the same as what's inside the plant itself, you just end up with a very regular looking cell. It won't be particularly swollen, but it won't be shriveled either. And that's because the movement in and out of the cell will be the same. Let's take animal cells now. I'm going to talk about a red blood cell, for example. So we're going to place our red blood cell in a salty solution. Because there's more water inside the red blood cell, similar to the plant cell, water will leave the cell and move outside, and the little cell will become all shriveled up again, and we call that crenated or crenellation. So our red blood cell isn't very happy at all. Now if we put our red blood cell in a pure water situation, you find that water comes flooding in. Why? Because there's much higher water potential, much higher water concentration outside of the cell. But the problem is this time that the water floods in, but because there's no cell wall, there's nothing to stop the ever-expanding red blood cell from bursting, so effectively it pops, which is a horrendous situation for our cell to be in because it's been destroyed. We call this process whereby it bursts, hemolysis, and it's a very dangerous situation for your body to be in. Now we need to talk about the cell cycle. So let's start by looking at chromosomes. So remember that your DNA is found inside chromosomes, which are found as pairs inside the nucleus of the cell, and this is why we say that the nucleus controls the activities of the cell. Now obviously throughout our life our cells don't just stay as old as we are, because you'd have an issue of some very old worn out cells not doing their job properly. So obviously they need to copy themselves and produce daughter cells, i.e. new cells, so we can carry on living, and that's where the cell cycle comes in. Now remember that during the cell cycle all that genetic information is copied, and that happens in mitosis, and then the cell divides in order to form two daughter cells, each with a full set of chromosomes. Now remember that in the cell cycle, in order to produce those genetically identical daughter cells, not only do we need to copy the genetic material, but we need to produce new organelles, so new mitochondria, new cytoplasm, new cell membranes, obviously, so that our daughter cell can be exactly the same as the original cell. Mitosis is the type of cell division which produces clones, so identical daughter cells, and these are needed in the growth and repair of organisms. We're going to talk about stem cells and differentiation. Now remember, a stem cell is a cell which is capable of becoming any other type of cell, so they're not specialised, and that's why stem cell research is so important now, because if you could harness a stem cell and get it to produce a hair cell, an eye cell, a skin cell as needed, then you'd have a very powerful tool. Now in animals, cells become specialised at a very early stage of their development, so when they're an embryo for example, and it is very difficult. You can't actually get adult cells to become any other type of cell. Plants are different to animal cells because they have stem cells which can constantly differentiate throughout their lives, and that's great for the plant because at any one point these cells can become any type of plant cell that the plant requires. These growing areas where mitosis occurs in a plant are found in meristems, which you find in the shoots and root tips. We're going to use the same thing in order to help you remember these definitions, because it's a hierarchy, they all build on each other. So remember, organelles are the small structures found inside cells. So an example could be mitochondria, it could be nucleus, it could be chloroplast. Now a cell in that case therefore is a group of organelles working together to perform the same function, and you're going to use those words to surround each of your answers. Now if that's not making sense, I'll move on to the definition of a tissue. And you're going to say it's a group of cells working together to perform the same function. And then if you move on to an organ, well, it's a group of tissues working together to perform the same function. And then lastly, you've got an organ system, which, as you might expect, is a group of organs working together to perform the same function. So we're going to leap straight in with testing for glucose. Now you want to use Benedict's reagent here, and you're going to heat your sample with some Benedict's reagent in a water bath. If glucose is present, you will see a brick red colour appear. If it's not present, then you'll just see the blue, which is the Benedict's reagent, and that is testing for glucose. Testing for starch is simplicity itself. You simply add a drop of iodine. Now iodine will turn blue-black in the presence of starch. It will stay like a yucky brown colour if there's no starch there. To test for proteins, you're going to add burette reagent. I don't know if I'm pronouncing it right, but I'll spell it right now. And what will happen if you've got protein is you will see a lovely mauve purple colour. Where are you rushing off to, Lyra? And then lastly, testing for fats, you're going to use the emulsion test. So first of all, start by adding some ethanol to your sample, which is a type of alcohol. Then you'll add some water. If fat is present, you will see a milky white emulsion or suspension, and it will just go cloudy effectively. I'm going to start by looking at the definition of digestion. Now digestion is the breakdown of large insoluble molecules into small soluble molecules so that they can be absorbed into the bloodstream. So we're going to start at the mouth. First of all, there's two types of digestion, physical and chemical. So physical digestion is literally just breaking large chunks of food into smaller chunks, but you're not actually changing their structure. Chemical is completely different. You're breaking down a large piece of food, but they'll actually become a new molecule. And really what we're talking about there is enzymes. So in the mouth you have physical digestion because you obviously have your teeth. However, at this point we get an enzyme released called amylase, and that gets released by the salivary glands. And what that's doing is it's breaking down starch into sugars. If you're not very happy with enzymes, check out my video on enzymes because I talk about them in much greater detail. So from the mouth we pass into the esophagus, the food pipe, into your stomach. So it reaches the stomach, and we know that the stomach is full of an acid, hydrochloric acid, which has a low pH. And what that's there for is to digest and break down bacteria to basically prevent us getting ill. At this point we have another enzyme released, and that's protease. And as the name, gosh, the cat hair's going everywhere, can't breathe. Anyway, so protease, as the name suggests, breaks down proteins into amino acids. So there's our large molecule proteins found in meats and pulses, and we're going to be making amino acids, which is small absorbable, is that a word? Molecule. At this point our pH is going to have drastically decreased, so when the stomach releases the food into the small intestine, the food's going to be very, very acidic, and its pH will be around 3. Now this is a problem because at this point we're having a load more enzymes being released. There's going to be more protease, more amylase, but also the addition of lipase, and they're going to come from the small intestine and the pancreas. Now this is problematic, the low pH, because what will happen is these enzymes will all denature, which means that they'll stop working, and that's because the pH is so far from their optimum pH, which will be around 7. So at this point we have another very important substance released, which is called bile. So what bile does is it neutralises the stomach acid and therefore increases the pH to around 7 so that these other enzymes can work properly. Bile is made by the liver. It's very important that you remember that. It's stored in the gallbladder and then it's released into the small intestine. So try and remember those 3 stages. As I've already mentioned, its first job is neutralisation of stomach acid, but it has a second very important role, which is emulsification. Remember, lipase breaks down fats into fatty acids and glycerol, and it's a pretty rubbish enzyme, which is why bile is important, because it increases the surface area. Right, so we talked about bile, we talked about our enzymes, and at this point, basically our food is pretty much all broken down, and we're moving into the small intestine, and we're going to get absorption of the digested food through the walls of the small intestine, so it can be transported around the blood. The walls of the small intestine are very specially adapted for their function, and they have these things called villi, which are like projections, and what they do is they increase the surface area for absorption to make sure it happens much faster. And on top of each villi there are microvilli, and they increase the surface area even further to make sure as much food as possible is absorbed through the walls of the small intestine. And then we get into the large intestine, and all that really does is absorb water and make sure that the end product is not nice and compact, but that it isn't like diarrhea basically. Gosh, it's a horrible topic. Let's start by our key definition of enzymes. Right, so an enzyme is a biological catalyst. That means that it speeds up the rate of a reaction without being used up. In terms of how an enzyme works, remember that an enzyme, you might have seen a picture, it's like a Pac-Man, and it has like a mouth, and we call that the active site. And what you have is you have a substance which fits into that specific active site on the enzyme. And what you find is that the substrate, we call that substance, breaks apart and produces a product. When the two are combined, what we have here is an enzyme-substrate complex. As with all science, it's really important that you use crucial keywords, things like specific, optimum, active site. Even if you're not sure of your answer in the exam, just literally vomit out all of these words onto your paper, and I promise you'll pick up some excess marks somewhere along the way, and it will really help bump up your grade. Finally, we need to talk about the effect of the conditions on enzyme controlled reactions, because enzymes are very, very fussy little things, and they really don't work very well if you don't treat them right. So the first condition we can look at is temperature. And with all reactions, if you have a very low temperature, you tend to find that the enzyme controlled reaction will occur very slowly. And that's just due to collision theory. Because if you have your enzyme and you have your substrate and they're just moving around really slowly, very lethargic, very low temperatures, you'll find that they don't really come together very often. So our active site and our substrate doesn't come together, and you end up with a very, very slow rate of reaction. What happens then is, as you increase the temperature, obviously these enzymes and the substrates will be moving around far more quickly, and you'll see a really nice increase in the rate of activity in the enzyme controlled reaction. And then right at the top, I'll find a graph to show you. What you see is there's an optimum temperature, and that really means, make sure you use the word optimum in the exam, it means the temperature at which the enzyme works best at. So pop down optimum temperature, you can normally read it off the graph, and it's right at the top. And then what you see is that after you've exceeded that optimum temperature, the rate of reaction literally drops off. And the word we use to describe that is denaturation. So the enzyme becomes denatured. And what that means is that the active site has changed shape, and therefore the substrate can no longer fit in. Because remember, it's like a jigsaw puzzle. They've got very specific shapes, and if the substrate can no longer fit in, then your enzyme's screwed. Make sure you never describe the enzyme as dying. You won't get any marks for that. The word you need is denatured. And then finally, the second thing we need to talk about is pH. Again, enzymes have an optimum pH. So, for example, in the body that will just be about 7, because pH 7 is neutral. However, certain other enzymes will prefer a far more acidic pH. So protease, for example, which is found in the stomach is full of hydrochloric acid. It has a pH of around 3. Therefore, protease's optimum pH will be around 3. As soon as you decrease it to 2 or increase it to 4, you'll end up with your enzyme rate of reaction dropping off, and therefore you get a very distinctive curve which looks like this. First of all, when we're talking about the heart, you need to know about the structure of the heart, you need to know the names of the vessels feeding in and out, and I'm going to help you with that. First of all is to remember that arteries carry blood away from the heart, always. So artery away, so that's a way to remember it, and veins carry blood towards the heart. Now the heart has 4 chambers that you need to know. Those are the atrium, left and right, and the ventricles, left and right again. So I've got a diagram here which is really simplified, but I promise that it does provide the information you need to know. Now, I find it easiest learning it like a box because I think some of the heart diagrams out there are very complicated, but I will show you one later on so you know that I'm not lying to you. Right, so if we draw a box and we divide it into 4, make sure the top 2 portions are smaller than the bottom 2 because the atria, which is plural, are smaller than the ventricles. So if you divide your heart into a box, the top 2 compartments are the atria, the left and right. Make sure you get the left and right sides the right way around. Underneath you have the much larger compartments of the heart, so that's why we've drawn our boxes bigger, and this is the left and right ventricle. Okay, so we're going to draw 2 vessels coming out of the ventricles because blood enters via the atrium, flows through the atrium into the ventricles and flows out of the ventricles via 2 vessels. What did I say earlier? The name of those vessels is the arteries, and we just need to assign their correct scientific name. So, blood that enters via the left atrium into the left ventricle will leave the heart in the biggest artery, the biggest blood vessel in the body, and that's the aorta. So make sure you get the spelling of that right. And if we go to the other side, blood which enters via the right atrium feeds into the right ventricle. That will leave via the pulmonary artery. And now I'm just going to talk a bit about naming. The word pulmonary means to do with the lungs. So that tells you that the pulmonary artery is feeding blood to the lungs. Because it's a circulatory system, it means it's a complete cycle. So it doesn't really matter where you start as long as you kind of end up back where you started from. So I'm going to start for some reason at the lungs. So what happens in the lungs? Right, well, the lungs are the organ where gas exchange takes place and oxygen enters our bloodstream. So what's going to happen? Oxygen is going to enter the blood via the capillaries and the alveoli in the lungs. It's going to enter the red blood cells and it's going to be carried in the pulmonary vein. Why? Because pulmonary means to do with the lungs. Vein, because we're bringing blood back to the heart. So it's going to be entering, and it's going to be entering on the left-hand side of the heart. Just a quick aside point. The 2 sides of the heart are split into oxygenated and deoxygenated sides. So as that sounds, it means that the heart is split into a side which contains oxygen and a side which doesn't. So the left-hand side always contains oxygen, and that's why I've drawn the vessels red, meaning that it's full of oxygenated blood. So our oxygenated blood enters the heart via the pulmonary vein into the left atrium. From there it flows into the left ventricle, and then it flows out of the heart via the aorta, where it goes around the rest of the body in order to deliver oxygen to cells which are respiring as they need it for aerobic respiration. So you can imagine that the blood then becomes deoxygenated because all the cells have taken the oxygen. That means that we need to pick up more oxygen. However, this blood can't just return to the lungs by itself. It needs to go via the heart in order to create that pump. So we have our deoxygenated blood returning to the heart, and this time, remember, it's going to enter via vein, and we're going to call this vein the vena cava, which is the main vein of the body. So it goes via the vena cava into the right atrium, flows from the right atrium into the right ventricle, and then it's going to leave the heart via an artery as usual, and this time we're going to call it the pulmonary artery. Why? Because it's to do with the lungs, because this blood will be returning to the lungs. The actual liquid portion of your blood is called plasma, and it's like a yellow liquid, and that carries dissolved things like sugar and urea and carbon dioxide back to the lungs where it's removed from your body. We've mentioned red blood cells. Obviously there are also white blood cells, and they play a really important role in your immune response, so basically protecting you from pathogens and microorganisms. So remember, they do that by ingesting pathogens or by producing antibodies which can destroy them. Lastly, you have platelets, and these are small fragments of cells, and what their role is that they clot the blood at the site of a wound. So if you cut yourself, it won't just bleed indefinitely because your platelets will come along and they will seal in that blood. Our nose and our mouths are connected to our windpipe, which we call the trachea, and then that splits off into two branches called bronchi. Then those branches become even more split and even smaller, and we call those the bronchioles. And lastly, we end in air sacs, which we call alveoli, and that's where gas exchange actually takes place. That's where oxygen leaves the alveoli and moves into the capillaries, and carbon dioxide leaves the capillaries and moves into the alveoli. And I'm just going to tell you now that that occurs by diffusion. So if we take oxygen, remember diffusion is the net movement of gas from an area of high concentration to low concentration. So that will be oxygen moving from inside the alveoli through its walls into the capillaries, and that is diffusion. Remember that the alveoli are adapted to maximise the amount of gas exchange that can take place. They're very thin, 1 cell thick, so there's a short diffusion distance. They're moist, they have a very large surface area. And is there one more thing? Yes, they have a plentiful supply of blood capillaries, so those are 4 marks for you right there. So let's just quickly remind ourselves of the equation. Remember, you need carbon dioxide plus water plus light energy, and then then we have our arrow, and then our products are going to be oxygen, that life-giving gas, and glucose. Hopefully my diagram will have flashed up and you'll see lots of different layers. We're going to start at the topmost layer. This is known as the waxy cuticle, and really that protects the leaf from the sunlight. Under that, it's a bit like a sandwich. We have an upper epidermis. Then we get into the really important part, and that's the palisade layer, or the palisade mesophyll. Look at those cells. They'll very closely resemble what you've learnt to be your regular plant cell, and that's because they are. And the crucial thing about these cells is they're packed full of chloroplasts. And remember, chloroplasts are full of chlorophyll, and that's where photosynthesis actually takes place. Underneath you have the spongy mesophyll, and surrounding that are air spaces, and that's really important because it allows our gases, oxygen and carbon dioxide, to circulate nicely. Some diagrams will include a vein, and all you need to worry about is that's where water enters the leaf, and remember that's entered via the roots in the xylem, X-Y-L-E-M. Okay, moving on, we have a lower epidermis, remember that sandwich I talked about, and then we get to the really important part, and that's the bottommost layer, and you will see a stoma and 2 guard cells. So the guard cells control whether the stoma is open or closed, because depending on the surrounding conditions, the plant will choose to close its stoma or open it. I'm just going to talk a little bit about transpiration right now. So remember, transpiration is the evaporation of water from the surface of the leaf, and that occurs through small pores called stoma or stomata. You will find that there are increased transpiration rates depending on certain conditions. The amount of water that leaves the leaf is based on the amount of water surrounding the leaf, because it's all to do with the diffusion gradient. So obviously water is moving from high concentration to low concentration. First of all, if it's dry, that means that there's very few water molecules outside the leaf because the air is dry, which means there's lots more water inside the plant, and therefore, based on what you know about diffusion, the movement of water will occur very quickly, so water will be leaving the stoma and you'll have high transpiration rates. Equally, if it's wet, if it's humid, there's a lot of moisture in the air, a lot of water molecules, so the difference between the amount of water inside and outside the leaf will be very little, so our gradient will be small and therefore transpiration won't be happening very much. Next, if it's windy, all these water molecules will have lots of energy and they'll be moving away from the leaf very quickly, and so you'll find that as the water diffuses out the leaf, it gets blown away quickly. And again, that means there's not very much water surrounding the leaf, so transpiration rates will be high when it's windy, and equally they'll be much lower when it's still air, because the water particles, the water molecules, won't be being blown away. Next up, if it's sunny, you'll find that transpiration rates are really high. Why is that? It's quite a complicated answer. If it's sunny, it means that lots of photosynthesis is taking place. What does photosynthesis require? It requires sunlight, but it also requires CO2. CO2 enters the leaf by the stoma. So in order to photosynthesize, the plant needs to open its stoma in order to allow that CO2 in. If you open the stoma, it means water will automatically start to leave. At night time, you find that the stoma tend to close because there's no sunlight, so there's no need to have it open, and no need for CO2 to enter, so you will find that less transpiration happens then. At high temperatures you find that transpiration rates are increased, and that's just because the kinetic energy of the water molecules is increased, and like the windiness thing, it means that they're just moving out of the stoma, out of the leaf much faster. And if you're in doubt with this and you can't remember anything I've said or anything you've learned in your lessons but you're in the exam, just think about the conditions which would help washing to dry and you'll certainly pick up some marks. Now, the last thing I wanted to talk to you about was the potometer. The potometer is used to measure the rate of transpiration. Here's a really terrible diagram of a potometer. So what's happening in this diagram is that water is leaving the leaves by transpiration, and therefore in order to replace that water you find that the stem draws in more water from the tube it's attached to, which is attached to a reservoir. And there's a little scale that you can use to measure how far the water's moved and how much water's been absorbed by the plant effectively. So you just read it off of the capillary tube, and that will tell you how much water has been absorbed. We assume that the rate of water uptake and the rate of transpiration are identical. Now, what sorts of things could you do to the leafy shoot in order to measure the rates of transpiration under different conditions? Well, you could put a bag over the plant in order to increase the humidity and hopefully see a decrease in the rates of transpiration and the rate of water uptake. You could use a hairdryer to emulate windy conditions, which would blow water away from the surface of the leaf. You could shine a light on the leaf. You could increase the temperature. So there's lots of things. However, in the exam, they like asking you about the setup of the equipment and any flaws and any ways you can fix them and how you can minimize error. So first of all, you want to avoid getting water on the leaves. So you might need to dry them if you accidentally splash them, and that's in order to prevent humid conditions being inadvertently given to your experiment when you're not actually interested in that. Second of all, you need to cut the shoot underwater, and that's to stop air getting inside the stem and preventing water movement because you'll have an airlock. You also want to cut the shoot at an angle to make sure it has a really good fitting into the rubber tubing. It's important to use Vaseline to seal the joints in order to stop air getting in places where it ought not to be. Finally, you use the capillary tube to magnify the amount of water uptake so you can actually see what's going on. First of all, we're going to define health. Now, health is the state of both physical and mental well-being. So what sorts of things might contribute to ill health? Things like stress, things like poor diet, so if you're not taking in enough vitamin D, enough calcium, enough iron, for example, and other factors which you can't control, such as your gender. So obviously men will get prostate cancer, girls won't, because we don't have a prostate. Women may get breast cancer. Men can get breast cancer, but very rarely, because obviously they have less glandular tissue there. Things like your ethnicity may affect how likely you are to get a certain disease. Right, moving on then, so key definition of a pathogen. Now, a pathogen is something like a virus, like a bacteria, and your definition is that it's a microorganism which causes disease. Crucially, know the difference between bacteria and viruses. Now, bacteria cells are quite big, they infect our bodies, they produce toxins and poisons that make us ill. Viruses are much smaller, they're not technically alive because they're so small, they're just a protein coat with a bit of DNA in them, and what happens is they infect your cells and then they actually hijack your cell's machinery to get the cell to work exactly as the virus wants them to, whereas the bacteria is self-contained cells. So if you're asked for like just some really small differences, you want to say that viruses are non-living and they're smaller, bacteria are larger and they are living. Okay, a more straightforward question now is how are pathogens spread? So you can use your common sense here. Think about all the times you got ill and why that might have been. So you could say droplet infection, and that's when someone sneezes or coughs on you, so keep those people away because they're disgusting. Some food and some drink will have a bacterial load, so something like undercooked chicken will have salmonella in it. Some infected water may have cholera in it. So again, be careful with what you're eating, make sure it's not undercooked, and make sure the water you're drinking is clean. Obviously there are things like STIs, so sexually transmitted diseases. They will be transmitted through direct bodily contact, and then there's stuff like drug users sharing needles that will help transmit things like hepatitis. So just be aware of all the different ways in which pathogens may be spread. Next up, we need to look at Petri dishes. The Petri dish is a little plastic dish which we use to grow bacteria on, because we're interested in that bacteria, how it grows and how we can actually prevent its growth using antibiotics. But in terms of preparing a Petri dish for inoculation, First of all, you need to get an inoculating loop, which is just a metal loop that you effectively swab the bacteria onto, but you first of all need to disinfect that loop to make sure there's no bacteria pre-existing on it, and you need to dip it into a blue flame of a Bunsen burner, again to ensure that it's totally sterile. At this point, you're going to swab it on the bacteria sample that you're interested in, and you're going to wipe it across the agar jelly that's inside your petri dish. Now, the agar jelly is a nutrient medium, which means that it's full of food that the bacteria can feed on. At this point, you're going to place the lid on the petri dish, and you're going to just make sure that some air can get into that petri dish to allow the bacteria to aerobically respire. And then you want to seal it with tape just to make sure that nothing spills. And that is how you're going to set up your inoculation and your petri dish. Another small weird point, which was also on the old AQA spec, is the role of Semmelweis. Now, he was a scientist, I think he lived in the 1800s, sorry, correct me if I'm wrong. And what was the important thing about him was that he recognised the importance of hand washing. Now, before that, doctors would just go between ill patient, covered in blood and gore, and go in, and if they were helping with a childbirth, they'd be there with their hands and everything, and they'd be transmitting these diseases, these pathogens, from one woman to the other. Semmelweis was amazing because he realised, no, you should not be doing this. Wash those hands, get rid of that bacteria that's stuck to your hands, and you will not kill these women. So he was an incredibly important guy. Now we're going to look at how pathogen spread may be prevented. You want to do stuff like sneezing into tissues. I know it sounds obvious, but don't go sneezing into your hands and then touching stuff. That's absolutely gross. And other things you can do when you're cooking is you can separate your raw and your cooked meats. You definitely shouldn't have raw chicken touching cooked ham. Not a good idea, because those pathogens will spread and make you very ill indeed. Next up, we are going to look at various different examples of viruses, and we're going to look at some protooctists, and you can call them protists, I think. I think protists might be easier. So yeah, we're going to start with the measles virus, so make sure you know that these diseases are caused by viruses, and you may have to specify that. Now, 100 years ago or something, measles was a very dangerous disease. It caused this huge rash, flu-like symptoms, and actually lots and lots of people died. However, since then they've developed a measles vaccine, which means that almost no one dies from measles virus anymore, so that's really, really good. Next up, we've got HIV. Now, HIV is a very famous disease. It stands for human immunodeficiency virus, and what happens is it's spread by sexual contact. It can be spread from mother to baby through breastfeeding. And what happens is you start with flu-like symptoms, and then over time it turns from HIV into AIDS. And when AIDS occurs, what you're doing is you're suppressing your immune system, because what happens is the virus affects all your white blood cells and stops them functioning properly, so they can no longer fight disease. So actually you die from a whole load of extra diseases that you catch more easily as a result of being infected with HIV. So yeah, not a good disease to get at all. Next up, we're going to look at tobacco mosaic virus, which I think I already mentioned. This is a disease which affects plants. It causes discoloration of the leaves, and it actually reduces the amount of chlorophyll available. Now remember, chlorophyll is found inside chloroplasts, and what it does is it reduces the amount of photosynthesis that takes place, and as a result that means less glucose is made, which means less amino acids, proteins and cellulose can be made, and also it means there's less glucose available for respiration, so less energy is made. So tobacco mosaic virus, not a good thing. Now we're going to look at some bacterial examples. So I'm talking about salmonella. I already said that you get that in undercooked chicken, particularly raw eggs sort of thing. You get really bad diarrhea and vomiting. I think anyone that's ever had salmonella poisoning really knows about it. And it is a bacterium. And you do find that most chickens are vaccinated against this disease these days, because it is such a nasty thing to catch. Now we're going to talk about gonorrhea, which is a very hard bacterial name to spell, and that is an STI, which means it's a sexually transmitted disease. And you get some really horrible symptoms with gonorrhea. You get discharge, and you need to know this, from both the penis and the vagina, and you will also find that there's pain when urinating, which means when weeing. Now, this did used to get treated very effectively using antibiotics such as penicillin, but more recently the bacteria has become very resilient to the penicillin, so we're finding that that's less effective, which is quite worrying because it's going to be hard to get rid of it. You need to use barrier methods such as condoms to prevent the spread of gonorrhea. Rose black spot is something else I've mentioned, which I know absolutely nothing about. Apparently it's a fungal disease, As the name suggests, it causes black spots on leaves, and those black spots will mean that there's less chloroplast, less chlorophyll, and therefore less photosynthesis. So everything I told you about the tobacco and mosaic virus applies here. So you'll have less glucose made, less amino acids being produced, less cellulose, and less respiration. So just make sure you're clear with that. And that was a fungal disease. We're moving on to our protist, disease now, which is malaria. Remember, that's spread by mosquitoes, and it's the female mosquitoes which come along, they bite you, and actually when they bite you, they infect you with malaria. Now, malaria kills millions, hundreds of millions of people. It's a terrifying disease, and it damages both your liver and your blood, and it sometimes can affect your brain. Now, the way in which you prevent the spread of malaria is actually by preventing those mosquitoes from biting you. So that could be wearing insect repellent, or using mosquito nets, or draining water sources which are stagnant, but I think that's more of a geography answer. Whew, right, I think we're done with the different types of diseases. So now we're going to look at how disease entry may be prevented. This is nice and straightforward, and there's lots of different ways. So first of all, the skin, the largest organ in the body, acts as a barrier. Secondly, you have stomach acid. Now, I know that the pathogen has to have entered your body to actually reach that stomach acid, but it hasn't actually been detected by your body yet, so it hasn't actually made you ill. So yeah, the hydrochloric acid kills pathogens. That's all you need to say for that point. Nose hairs trap pathogens, your third point. And lastly, the cilia in the trachea, they trap the pathogens and waft it up to your throat, where it can be swallowed and destroyed by stomach acid again. So those are the 4 ways in which pathogen entry is prevented. Right, next up we're looking at, once those pathogens have entered the body, what happens in order to actually destroy them, and once they've made us ill, how do we actually stop them making us ill? Because after all, most of the time we're not ill for weeks and weeks on end. And that's all due to your white blood cells. Because first of all, you have white blood cells which engulf the pathogens, which effectively means that they eat them. So you write they engulf pathogens. Second type are called lymphocytes, and they produce antibodies, and they recognize the antigens on the pathogen. You need to look at my white blood cell video if you're not understanding this. And they produce antibodies which destroy the pathogen, and they also produce antitoxins. Right, next up, how the vaccinations work. So remember, if you go to some tropical country, you may need to get a vaccination, and all that's happening in this case is a dead or weakened form of the pathogen is injected into your body. Your lymphocytes produce lots of antibodies against that pathogen, and what happens therefore is the next time you go to the country or whatever, you get infected, well, you've already got a massive store of antibodies ready to attack them, so you can't actually get ill in the first place. So that's why vaccines are so important. Oh, AQA, why is it so random? Oh, they're now wanting us to discuss aphids. Now, aphids are little green insects, and what they do is they bite into the phloem of a plant. Now, a phloem is a tube which transports sugars around the plant. So obviously they bite into that phloem to access the sugar so they can feed. So what issues does that cause? Well, obviously there's less glucose available, so less respiration can take place, less amino acids can be made, less protein, and less cellulose. So they do cause a lot of damage. Define chloriosis, that's just the yellowing of leaves due to lack of magnesium ions. You do need to know the reason why plants need magnesium. Plants need magnesium to make chlorophyll. Plants need nitrates in order to make amino acids and protein, and they may very well ask you that. Next up, how may plant diseases be treated? So we may use pesticides, which obviously kill pests such as aphids. You can use antifungal treatments to destroy things such as black spots on roses, and obviously you could cut away the diseased areas. So yeah, we're going to use fungal treatments, we're going to use pesticides to kill aphids, and you're going to cut away the diseased areas. How may plant diseases be identified? Well, you can send them off to the lab, or you can use home testing kits which contain monoclonal antibodies. The last 2 things I wanted to cover is how plants actually prevent invasion by pathogens. Now, lots of different ways. First of all, they have a waxy cuticle which prevents pathogen entry because it's very waxy, it's very thick, and pathogens find it hard to cross. Next up, lots of trees have thick bark, and obviously thick bark is very difficult to get through. You have cellulose in the cell wall, which I've already mentioned a lot, and that actually strengthens the plant cell and again helps prevent pathogen entry. Another thing that plants do is they'll produce poisons and toxins and lots of chemicals which make them very unappetizing and make it difficult for the pathogens to actually affect them. The last thing I wanted to mention is that plants have this clever thing where deciduous trees lose their leaves in autumn, and the great thing about that is if there's any pathogen which has been infecting the leaves, it means that they actually drop those leaves in autumn and therefore they lose the pathogen as a result. Lastly is, how do plants prevent animals eating them? Because remember, herbivores are animals which just eat plants. So plants will make themselves very unappetizing to these animals by growing lots of thorns, lots of hairs, which are very uncomfortable and unpleasant to eat. And they do something called mimicry, which is they'll actually copy the leaf pattern of a more poisonous plant, so the animal's scared to eat the plant in case they get poisoned. So that's a really clever way in which the plants will prevent eating. And lastly, something like mimosa. If you've ever touched mimosa when you're on holiday, it's a special leaf that when you touch it, it curls up and looks really thin and unappetizing and unappealing. And plants will actually shrivel up and curl away, and animals can't bother to eat them because they think there's no plant to eat. Now remember, when we're talking about the immune system, we're talking about, remember what happens when you have bacteria and they enter your bodies and they make you ill. And obviously you get sick, but for most people you don't stay sick for like years and years and years. So we're talking about how our body actually fights against those bacteria, and that is your immune system, and it's specifically your white blood cells, and they have some fancy names. So, for example, if a cold virus enters your body, what happens is white blood cells called lymphocytes come along, and they recognize specific molecules on those cold viruses, and we call those molecules antigens. So a very specific lymphocyte will come along and it will release antibodies which will go and attack that cold virus, and it will help destroy it, and that's how our immune system actually works. Now the way vaccinations work, remember that's what happens if you're going somewhere tropical on holiday, or as a kid you'll have probably had a lot of vaccinations, maybe against HPV, meningitis, that sort of thing. And the way vaccination works is it kind of speeds up your immune system. You have an injection, and what they're injecting is a weakened or dead form of the pathogen. Now remember, the pathogen is the disease-causing organism. So in this case it's the flu virus, it could be the polio virus, can be many different types of pathogen. But what a vaccination is it's a dead form, dead or weakened form of that pathogen. Why does it need to be dead or weakened? Well, obviously you don't want it to enter your body and make you ill, because that would make that vaccination completely pointless. So they inject it into your body, very specific lymphocytes will come along, they'll recognize the antigen on that pathogen, and they'll start releasing antibodies. And some of those lymphocytes then turn into memory cells, and that's a really clever, cool thing, because a memory cell literally memorizes that particular pathogen, so whether it's the flu virus or the polio virus. So that if you happen to come in contact at some other point in your life with that polio, with that flu virus, your body already has memory cells which are waiting, and the moment that that polio enters your body, you've got massive numbers of memory cells that will start booting out lots and lots of antibodies. Those antibodies will go and attack and actually stop you getting ill in the first place. So vaccinations are really, really important. Herd immunity is just a term describing when a large number of people get vaccinated against something, because something like measles virus will need a large number of people to infect. So it will infect a person who will then go and infect another person that will go and infect another person. And that's how the measles virus is spread. Now obviously, if you go and vaccinate loads of people against the measles virus, there are going to not be enough people for the measles virus to spread properly between. And that's how you actually get herd immunity against something like measles. It basically means when a large number of people are vaccinated against a specific disease. Now the types of vaccinations you might have heard of are things like MMR, which is measles, mumps and rubella, and they may ask you that. And I've already mentioned you may be vaccinated against something like polio. If you've been on holiday, it might be tetanus or cholera, typhoid, all pretty nasty diseases that need vaccinating against. Now we're going to talk about the different types of drug. Now remember, a drug is just a chemical which has an effect on the body, so it may be a good drug or a bad drug. So things like coffee contain caffeine, which is a drug because it stimulates you, it increases your heart rate. Then you've heard of things like heroin and cocaine, they're bad drugs, they have bad effects on your body. But a drug in its simplest term is a chemical which has an effect on the body. Now if you get ill, you may take things like aspirin, paracetamol, Nurofen, ibuprofen, and what these are tablets which reduce the symptoms of whatever it is you're suffering from. So they don't actually go to kill the pathogen that you're suffering from. What they do is they alleviate the symptoms, which means they may reduce the headache you're suffering from, they may get rid of some of those snuffly noses, help clear your sinuses, that sort of thing, but they're not actually killing any of the pathogens. If you want to kill a pathogen like a bacteria, you're going to have to use an antibiotic, and antibiotics only work on bacteria. They do not work on viruses. So I've already described one problem with antibiotics, which is that they're only effective against bacteria. The second problem is that increasingly we're seeing antibiotic resistance. And that's what happens when we don't complete the course of antibiotics that we're taking, if we over-prescribe them, because you find that you'll get some strains of bacteria which are actually resistant to the antibiotic, and then before you know it, all the new bacteria that are being made are resistant. So back in the day when a certain antibiotic would work, you're finding later that it doesn't work, and obviously that's going to cause massive issues if we don't have antibiotics that work. Let's talk about how we actually go about developing drugs then to treat ourselves. So in terms of what we're after from a drug, obviously you want it to be effective, because what's the point of taking a drug if it doesn't actually work? You want it to be safe, so you don't want to have it causing any adverse effects on your health, because that wouldn't be great. You want it to be stable, and what that means is that it needs to be easily stored. You don't want a drug that can only be taken within like a week and then goes off in the cupboard and starts causing like problems. You want it to be nice and stable. And lastly, the least obvious one, it needs to be successfully taken into your body, so easy to deliver, whether that's drinking it or having it injected, and it needs to be able to be removed from your body too. I don't know if you know this, but lots of drugs come from plants, and lots of these plants are found in the rainforest, and obviously you can't get some chemical from a plant and then just give it straight to someone, because you don't know the effects it will have and it may kill them. So there are lots of stages involved in the development of a drug. The first stage is preclinical trials, and that's when they test a chemical or a drug out on tissues or cells or other animals, and that's where animal testing comes in, and that's why lots of people have lots of problems with animal testing, and I understand that. Once they've tested it out on the animals, on other cells, they will then start testing them out on human volunteers, and we call that clinical trials, and they'll start by giving human volunteers very, very small amounts, so that if there are going to be bad effects, hopefully they're not going to be too dangerous, and then they'll increase that dose, the amount that they give the person, bit by bit, until they're happy that it's the right amount. When you're talking about actually doing trialing, there's something called a double-blind trial, and that's when neither the doctor nor the patient know whether they're being given the drug or not. And that's essential, because a lot of times if a patient is given a drug and told that it's really going to work, they actually kind of get themselves better through their own mental state of mind. They'll be like, wow, I've been told this drug will work, so I'm going to get better because of it. And that's obviously giving you a false positive result, because it's not necessarily the drug doing that. So a double-blind trial is when neither the patient or the doctor actually giving the drug know whether the patient's had the drug. Because the other thing they could be given is a placebo, and a placebo is just a tablet that's like made out of sugar that has no drugs in it. So some patients will be given a placebo, some will be given the drug, and then you'll look at the results to try and work out how effective that drug is. Now we're getting onto the tricky topic of monoclonal antibodies. So what we're talking about here is a new development, it's a new technology, and we need to first of all understand what the word hybridoma means. And all that means is it's a new cell type formed by combining mouse cells, human cells, and cancer cells in order to form a new type of cell. So I've already told you that lymphocytes will produce antibodies which will go ahead and destroy pathogens, but the problem with lymphocytes is they don't actually divide and grow. So in an ideal world, you'd get a load of someone's lymphocytes, cause them to divide to make loads of antibodies, and then go around treating people using those antibodies. But I've just said that lymphocytes don't divide. So what we need ideally is a type of cell which divides rapidly, and in this case we know that cancer cells divide very rapidly because they go ahead, they form tumours. That's a really terrible thing about cancer cells, is these tumours that grow and obstruct parts of our organs. So if they're in our brain or our heart, that's where all the issues arise from with cancer. So if you can think you could get a lymphocyte that's producing all those awesome antibodies that will go and kill stuff, but then you've got the cancer cell which has the ability to copy itself and divide lots. Imagine if you combined them, then you'd have the perfect situation where you're going to generate lots of antibodies that will go ahead and kill stuff, but won't be cancerous and therefore won't cause any problems. And that's really what this monoclonal antibody stuff is all about. Now, what does the term monoclonal antibody mean? Well, it's just a protein which is made to target specific cells. So the crucial thing here is they're very specific. Now, they found that mice lymphocytes, so that little mouse, they found that lymphocytes inside the mice are amazing at producing these monoclonal antibodies. So they combined it with the cancer cell in order to form this hybridoma, which I've already mentioned, and then that could go ahead producing all the antibodies needed to destroy dangerous cells. I mean, the obvious problem with this is that they come from mice, and obviously our bodies are going to recognize mice cells as being foreign, and then our immune system will try and attack those mice cells. So what they then did was they combined it with human cells in order to prevent the rejection issue, and that's why the hybridoma is a combination of mouse, human and cancer cells. So how can monoclonal antibodies be used? Well, firstly, pregnancy testing, because there's a hormone called HCG which pregnant women make. Now what happens is the monoclonal antibody can actually bind to that hormone, and then it gets expressed in urine, which can then be detected. So it's actually impossible to get a false positive result. You will not get a false positive result. You will definitely be pregnant if you get a positive result. So I think that's quite an essential life point right there. The second thing that you can use monoclonal antibodies is for detecting disease, because it can actually bind to disease cells, and that's used to detect prostate cancer in men, for example. And lastly, you can use monoclonal antibodies to detect levels of hormones. So an example of this is, you know how people donate blood, because blood donations are really important, but obviously you don't want someone with the disease AIDS or HIV donating blood, because that could go and infect the poor people that the blood's being given to. So monoclonal antibodies can actually be used to screen that blood and see if there's any HIV in there. Really, really good stuff. Now, how can monoclonal antibodies be used to treat cancer? Because really that's what we're after. That's the really amazing role that they perform. So first of all, they can help carry toxic drugs to the cancer cells, and therefore those toxic drugs can go to work on the cancer cells without affecting our normal cells. Secondly, the monoclonal antibodies can help trigger our immune system to actually go and attack those cancer cells, whereas before it might not have been doing that. And thirdly, those monoclonal antibodies can block the receptors on cancer cells, and those receptors are needed to help that cancer cell divide to form a tumour. So if it blocks the receptors, they can't divide anymore, which is obviously essential to prevent tumour growth. Now we need to look at the advantages of monoclonal antibodies. The first thing is that they have no effect on healthy cells, and that's crucial because a lot of cancer treatments such as chemotherapy and radiotherapy do go and damage a lot of healthy human cells, which is obviously not what we're wanting. The second thing is that the monoclonal antibodies can be used to treat a wide range of conditions. However, sadly, there are disadvantages. That is, some side effects can arise from using monoclonal antibodies. And actually the technology involved has been tricky to develop, much trickier than they first imagined when they first started developing this. So yeah, what is a non-communicable disease? Well, it is a non-infectious disease which is caused by other things such as your environment or your genes. Now, there are lots of different risk factors which increase your chance of getting a particular type of disease, which I'm going to go into now. So first of all, your genes. So lots of people out there can't get cystic fibrosis because they don't actually inherit those genes. So it's not a spreadable disease, but it is an inherited disease. Other risk factors include your sex. So obviously women are more likely to get breast cancer, men will get prostate cancer. There's things like your lifestyle choices. So if you choose to smoke, obviously you'll increase your chance of getting lung cancer. If you drink too much, you'll increase your chances of getting liver cancer. I think Martin's going to shout at me because I'm very small. He keeps telling me off for being really small in my videos. Is that zooming in? Sorry Martin, looks like I'm really small. So yeah, lifestyle choices for sure, and other things such as ionizing radiation. So that could be UV light from the sun, for example. Right, let's now look at the horrible topic of cancer. Now you need to know all these different terms and what they mean. Let's start by looking at what a tumour is. Well, it's a mass of abnormal cells, so effectively a big lump. Now tumours can either be benign or malignant, and benign tends to mean the good one, if there is a good type of tumour to get, and malignant's the one that gets people quite scared. So we'll start by looking at the benign tumour. Now the thing about these is they just grow in one place, so they're contained to one area of your body, and that's a good thing because it won't actually tend to be super harmful because it's literally just a lump. The times when it can be tricky is if that lump or that tumour grows somewhere like your brain or your heart, because there's nowhere for it to really grow out into, and it's that pressure of it building up and putting a force on the surrounding tissue that actually may cause you a lot of harm. But generally speaking, benign tumours are okay. Now, it's malignant tumours that, like I've already said, get people worried, because these are tumours which spread from one part of the body to another, and they form secondary tumours wherever they end up, because you can imagine that a part of a primary tumour may break off, get transported in your bloodstream or your lymph system, and it can end up being deposited at other parts in your body, and it's there when it starts growing, obstructing blood vessels, etc. That's where it causes the problem. So yeah, malignant tumours are terrible, terrible things. Now, the reason people get so scared of cancer is because it's actually really hard to tackle, because all it is your actual normal body cells just dividing in an uncontrolled way. It's not like they're a foreign pathogen, it's not like they're a bacteria or virus that our immune system can go and attack because it recognises it as being foreign. The problem with cancer cells is that they're just our normal cells that have just gone out of control. So our white blood cells and our lymphocytes and our phagocytes aren't actually going out to detect them, which is why they can remain undetected for years sometimes. Now, there are two main ways in which we treat cancer. One is radiotherapy and one is chemotherapy. So as the name suggests, chemotherapy involves chemicals, and they're targeted at the cancer cells. And what they do is they cause those cancer cells to self-destruct, i.e. to kill themselves. However, chemotherapy makes people very sick, has lots of horrible side effects. Some people's hair falls out, some people get very, very sick, nauseous, and it's really hard to target it specifically at the cancer cells, so it can cause a lot of damage to surrounding healthy cells. Radiotherapy is slightly different. This time we're using radiation, which we fire at that tumour, at that cancer, and we try and destroy the cells that way, and we try and stop them dividing by mitosis. But again, healthy cells can be affected, which is why neither of these ways are ideal solutions, and this is why we're looking at monoclonal antibodies. We're going to talk about the different risk factors to do with getting diseases. So we're going to be looking at smoking now, really, and why smoking can lead to so many problems with your health. And that's because cigarettes contain lots of horrible, horrible chemicals. First of all, they contain tar, which is a black sticky stuff, which is a carcinogen. Now, a carcinogen's definition is that it's a cancer-causing agent. So what tar does is it causes some of those cells inside your lungs to divide abnormally, and some of those can become tumours. It also coats the inside of your lungs with that black sticky stuff, and if you've ever Googled a smoker's lung, you'll see exactly what I'm talking about. The lung actually turns black. It's really gross, whereas a healthy lung should be a nice light pink colour. Cigarettes also contain nicotine. Nicotine is the addictive drug that is the reason why people who smoke find it so hard to give up. Nicotine makes your blood a bit more viscous, it makes it thicker, which means that you're more likely to get high blood pressure, and that can lead to issues to do with your heart. So that's a causal factor towards heart attacks, so terrible stuff, nicotine. You've got carbon monoxide, which is a type of gas. Now, carbon monoxide combines with the hemoglobin in your red blood cells, and it stops your red blood cells transporting oxygen. So obviously you're transporting less oxygen around your body, which is obviously a terrible thing, and you will get very short of breath. You can get emphysema, which is a horrible disease whereby your alveoli, which are the air sacs in your lungs, they can get destroyed, they can expand, which reduces the surface area for gas exchange. So emphysema is a very nasty disease. You may have heard that cigarette smoke can paralyse the cilia, which are these special cells inside your windpipe, your trachea. What they do is they waft, and they help remove mucus and bacteria from your lungs. Well, if they're paralysed, then that bacteria is just going to sit there, and that leads to infection, where things like bronchitis come in. So yeah, all in all, terrible, terrible things to smoking. I'm trying to think of other things. Premature aging, we know people, they get the lines around their mouth from the sucking of the cigarette. You can get yellow tans from the nicotine, so it's a pretty unpleasant habit, really. And then obviously, I've already mentioned this, but like massive link between smoking and heart attack. And talking more about heart attacks, there's lots of risk factors. There's lots of different things you can do which will increase your risk of getting a heart attack. One will be smoking, one will be drinking too much. Another thing will be if you become obese and you're eating too much, because you're more likely to get diabetes type 2, which has a very strong link towards cancer. If you don't exercise enough, if your blood pressure gets too high, all of these factors interrelate and may lead to heart attacks or strokes. So let's talk a bit more about diabetes type 2. Diabetes type 2 is due to poor diet, and what happens is your pancreas stops responding to the insulin, so you can't lower your blood sugar levels. People with diabetes can end up in quite serious trouble. They can lose their eyesight, they can lose the ability to walk, and like I said, it is a massive risk factor towards heart disease. Now we're looking at drinking too much. So people who are alcoholics, they may cause their liver to become cirrhosed or get cirrhosis, which is where portions of the liver cells die effectively. You may develop liver cancer. People who drink serious amounts may actually change the structure of their brain, and the texture of it can become very pulpy and mushy. So obviously they're not going to be able to process things properly anymore. And the problem with alcohol is it is an incredibly addictive drug. Some say it's more addictive to things like Class A drugs such as heroin and cocaine. And the other thing is it's socially acceptable to drink, so it's quite a hard thing because it's so readily available. And yeah, I think it's quite hard for people to realise if they have got a problem, because it's quite normal to go out for lots of drinks. The real issues arise with alcohol is in pregnant women, because if you drink a huge amount while you're pregnant, your baby can develop fetal alcohol syndrome, which means that it could be born deformed. It could have alterations in its limb structure, its jaw, for example. You've got issues with heart problems, kidney problems, liver problems. So remember, we have our green plant and it needs to absorb carbon dioxide and water, and the water, remember, comes in at the roots. So the two products produced are oxygen and also glucose, which is what the plant's actually after. If they ask you for the balanced symbol equation, remember it's sixes, sixes, sixes. So for example, we need 6 CO2s plus 6 H2Os, which, remember, that's 6 waters, and that goes to C6H12O6, which is the formula of glucose, plus 6 O2. Remember that photosynthesis is an endothermic reaction, which means it takes in energy. Now, if they ask you about how the leaf is adapted for photosynthesis, you're going to mention the following things. First of all, it's broad and flat, which means it has a large surface area to trap that sunlight, to absorb that sunlight onto the leaf. It's thin, and that means that diffusion distances, so things which are diffusing are things like the gases, that is a nice short distance, so they'll diffuse nice and quickly. You've got air spaces within the leaf which will enable those gases to diffuse again. Remember, the palisade layer has lots of chloroplasts, and those chloroplasts contain chlorophyll, which trap that sunlight, and chloroplast is actually where photosynthesis takes place. Within the leaf, you've got a vein, and the vein brings up water from the roots and actually supplies the leaf with all its water needs. You've got stomata and guard cells lastly. Remember that they're super important. The stomata is a pore inside the leaf which actually allows carbon dioxide into the leaf and water to leave. And those guard cells surrounding the stomata actually control whether the stomata is open or not. So obviously, if it's open, CO2 can get in, and if it's closed, it cannot. Now we're moving on to the limiting factors which affect photosynthesis. So there's 3 ones you need to remember. That is carbon dioxide, temperature, and light intensity. All 3 of these things can limit the amount of photosynthesis can take place. Let's start with the most simple one of those, temperature. Obviously, if you have low temperature, then those reactants involved will be moving slowly. It's a bit like the effect of temperature on enzymes. So the particles don't come together fast enough, and therefore the whole reaction slows down. So low temperatures will lead to low rates of photosynthesis. Obviously, at very high temperatures, you'll see that the enzymes involved in photosynthesis will be denatured, so you don't want to raise that too high. In terms of carbon dioxide, obviously low amounts of carbon dioxide will limit photosynthesis, because actually it will slow down how much photosynthesis can actually take place, because without CO2, no photosynthesis. Same with light. Light powers the whole process. If you've got low light levels, then you're going to see low rates of photosynthesis. And remember that all of these 3 things interact with each other, so you could have the most amount of CO2 ever, but if you have a low temperature or low light level, you're not going to see that much photosynthesis. You could have the most amount of light ever, but if you've got low CO2 levels, again, we're limiting the amount of photosynthesis. Just going to quickly touch on experiments involving photosynthesis, and they tend to use something called Elodea, which is pondweed, to measure the rates of photosynthesis. And the way they'll do this, remember, hopefully you've been shown this at school, is they'll get a piece of that pondweed, that Elodea, and because it's pondweed, it lives in water, so you pop it into a beaker full of water. Because it's photosynthesizing, we've already said that oxygen will be given off, and you can actually see those bubbles being released by the Elodea. So sensibly, you can actually count the number of bubbles that appear within a certain time frame, so something like 60 seconds is good here. So if you count the number of bubbles produced in 60 seconds, you can kind of get an estimation of how quickly photosynthesis is taking place. And if you alter the conditions that the Elodea is experiencing, then you can actually alter the rate of photosynthesis in that way. So one popular way is to alter the light intensity. So you may or may not have got a lamp popped at 10 centimeters away from that pond reed, count the number of bubbles, 20 centimeters away, count the bubbles, 30 centimeters away, count the bubbles. So in terms of your independent variable, remember that's the variable you're changing. So in this case, it would be the light intensity, i.e. the distance the lamp is away from that LODIA. The dependent variable is what you're measuring, so you're measuring the number of bubbles in 60 seconds. And the control variable is everything you need to control to keep it a fair test, to keep it the same. So obviously you need the same species of pondweed, so LODIA in this case. You need to have the same length of pondweed, because obviously a longer pondweed will photosynthesize more. You need to have the temperature the same, the pH of the water the same, and technically the concentration of carbon dioxide the same, although that will be quite hard to control. So let's start with aerobic respiration. Aerobic meaning using lots of oxygen, as opposed to anaerobic respiration, which means respiration without oxygen. So let's first of all take the equation for aerobic respiration. So we're going to be taking oxygen into our bodies, so it's oxygen plus glucose, which we've eaten in our food, an arrow, and then you're going to produce carbon dioxide, which you'll breathe back out, lots of water, which you're breathing out as water vapour, and finally energy, which is the whole reason we're doing this. So, like I said, it occurs in the mitochondria of cells, and it's our way of releasing lots of energy. Now, what do we need to use that energy for? Well, we need to use it to build up smaller molecules into large ones, so for example building amino acids into proteins. We need it to maintain our body temperatures, because as mammals we know that we are warm-blooded. And lastly, we need energy to help our muscles contract. There's lots of other things we need energy for, but those are the three main ones. However, sometimes we can't get enough oxygen into our bodies to allow us to aerobically respire, and this time we'll choose to do anaerobic respiration. Some such example of this could be when you're sprinting, like the 100-meter race at school. You just can't breathe fast enough. The advantage of this is that you can use it when there's not enough oxygen around, but the problem is that it releases far less energy in comparison to aerobic respiration. And you may have seen also that glucose, rather than being broken down immediately into carbon dioxide and water, is instead broken down into lactic acid. Now, lactic acid is toxic, and it leads to that horrible stitchy, crampy feeling, so it leads to muscle fatigue and cramping, and that's kind of problematic. So what we need to do is take in even more oxygen into our body post-run, post-race, in order to break down that lactic acid into carbon dioxide and water. And that extra amount of oxygen that we need is something we call the oxygen debt. And all it does is it means that it's allowing way more oxygen into our bodies to allow that lactic acid to be broken down, so it stops causing fatigue. They like to ask you a little bit about the liver if you're doing the higher exam board. So remember, it's a big reddish brown organ that sits around here. It's involved in an awful lot of metabolism roles. And the great thing about liver cells is they're able to regenerate and grow very readily. So what role does the liver have? First of all, it detoxifies, and that includes breaking down alcohol that you've drunk. Next, it gets breakdown products and passes them into the blood so they can be excreted by the kidney. And lastly, it breaks down old, worn out red blood cells and helps to store that iron stored inside them until it's needed to make new cells. Really hope you found this video helpful. It was such a nightmare to make. It's taken me, I feel like, hundreds of hours. So yeah, please sub and give it a like if you want a physics video. See you very soon, guys. Bye.